This is the Dive Into Discovery podcast, where curiosity meets insight, and every conversation uncovers a new layer of understanding.
You know, when we talk about DNA, we often think about genes, right? Those blueprints for building proteins. But what about all that other stuff, that vast majority of our DNA that doesn't code for proteins? You know, that mysterious 98% that we used to just call
junk DNA.
Yeah, junk DNA. Well, you've sent us a ton of research on this topic, and well, let me tell you, the story of junk DNA is a lot more interesting than the name suggests.
It really is. It's a great example of how scientific understanding kind of evolves over time. You know, the term junk DNA itself, I mean, it kind of reflects this outdated view we had, like back when we just didn't have the tools or the knowledge to really decipher all the complexities of the non-coding genome.
Yeah, it is kind of humbling in a way, right? To think that we just labeled this massive portion of our DNA as junk just because we didn't get it. So for our listener, let's break it down a bit. What exactly are we talking about when we say junk DNA?
Well, essentially, we're referring to the stretches of DNA that don't directly contain instructions for building proteins. So those non-coding regions, they were initially thought to be useless, but more and more research is revealing that they actually play a critical role. They're involved in regulating how our genes function, almost like a complex control panel, you could say.
Oh, yeah. I was reading about that. It's fascinating how these non-coding sequences can determine when and where genes are turned on or off. Is it as straightforward as like an on-off switch, or is it more nuanced than that?
It's definitely more nuanced than that. It's less like a single switch and more like, you know, one of those sophisticated control boards, you know, with dimmers and timers and all sorts of fine-tuning mechanisms. We're talking about elements like enhancers and promoters. These things can amplify or they can suppress gene activity, making sure that the genes are expressed at the right time, in the right cells, and the right levels. And this regulation is crucial for everything. I mean, from, you know, way back in embryonic development all the way to preventing diseases.
So it's not just about whether a gene is on or off, but like how much, when, and where it's expressed. And these non-coding sequences are basically conducting this whole intricate orchestra of gene expression.
Exactly. And it's not just these regulatory elements. Junk DNA also produces this fascinating cast of characters, you could say. Molecules like microRNAs and long non-coding RNAs.
Okay, those names ring a bell, but just remind me again, what do those things actually do? It's like a whole new vocabulary to learn.
Well, think of them as like the cellular managers, you know, kind of like orchestrating all those complex processes within our cells. So take microRNAs, for instance. Those are tiny molecules that can bind to messenger RNA, which is, you know, the molecule that carries the genetic code from the DNA to the protein-making machinery. And by binding to messenger RNA, microRNAs can actually fine-tune protein production, essentially like silencing or tweaking the message in a way.
So these tiny molecules are like little editors shaping the final product. That's
Yeah.
amazing. And what about those long non-coding RNAs, those LNC-RNAs? What's their deal?
Well, they're kind of the bigger players in this cellular drama. LNCRNAs can act like scaffolds, you know, bringing together different proteins, or even as decoys, intercepting molecules that might be, you know, interfering with cellular processes. They're involved in a whole bunch of activities, from cell development and differentiation to even the progression of diseases.
It seems like we're only just starting to really appreciate the complexity and importance of these non-coding RNAs. And then you've got transposons, those jumping genes that sound like something straight out of science fiction. What's their story?
Transposons. They're like these mobile genetic elements. They're capable of moving around the genome, almost like, imagine sentences rearranging themselves within a book. Now, the implications of this are fascinating. Some scientists believe that transposons have actually played a significant role in evolution.
That makes a lot of sense. If these transposons can move around and insert themselves into different parts of the genome, that means they could potentially create new combinations of genes.
Leading to new traits, right? Exactly. Like, think about the ALU element. That's a type of transposon that's found in primates. And get this. It's estimated that ALU elements actually make up about 10% of the human genome.
Wow, 10%. That's a huge chunk of our DNA.
It is. And while their exact role is still being debated, there's evidence to suggest that they've actually contributed to the evolution of some uniquely human traits.
10% of our genome, it really highlights how much of our genetic story is still being written, But it's not just about those individual elements like transposons, right? We're also seeing that some non-coding sequences are remarkably similar across different species.
That's right. These are what we call conserved sequences. And they're another piece of the puzzle. The fact that these stretches of non-coding DNA have remained largely unchanged over, vast evolutionary distances, it suggests that they must be doing something incredibly important, like really, really important.
It's like a message passed down through generations, right? A whisper from our evolutionary ancestors. And I was reading that a lot of the differences between human and chimpanzee DNA, even though we're so closely related evolutionarily, a lot of those differences actually lie within these non-coding regions.
Yes. And that just strengthens the idea that non-coding DNA plays a really crucial role in defining the unique characteristics of each species. It's a reminder that evolution isn't just about changes in the genes themselves, but also in how those genes are regulated.
This is blowing my mind. It seems like everything we thought we knew about DNA is being challenged. But if these non-coding regions are so vital, why do we see them conserved across species? What evolutionary advantage could that possibly offer?
That is a great question. And it's one that scientists are still trying to figure out. One possibility is that these conserved non-coding sequences are involved in some really fundamental processes. Processes that are essential for basically all life. Processes that have remained largely unchanged over millions, even billions of years of evolution.
It's humbling to think about those deep connections we share with all living things, right down to the level of our DNA.
It really is. It speaks to the power of evolution. How it shapes and refines the blueprints of life, basically. But the story of junk DNA doesn't end there. You see, we've been focusing on its role in regulating genes, but it's also crucial for maintaining the actual structure of our chromosomes.
Wait, hold on. I always imagined DNA as this long, straight ladder. How does it have a 3D structure?
It's more like, imagine a tangled ball of yarn, like intricately folded and packed within the nucleus of our cells. And this is where non-coding DNA steps in. It acts like scaffolding. It helps to kind of organize and compact that tangled mess into a functional form.
So it's not just about, controlling the genes, but also about keeping everything organized within the cell.
Exactly. And a key part of that organization are the telomeres.
Oh, right. The telomeres, those are the protective caps at the ends of our chromosomes, right?
Yeah.
I've heard they're linked to aging.
Yes. They're made up of repetitive DNA sequences, which, would technically fall under that jump category.
Yeah.
But telomeres are absolutely crucial. They prevent damage to our genetic information, and they ensure proper replication of our chromosomes. And you're right, they do shorten as we age, which is why they've become such a focus of research into aging and age-related diseases.
Wow. So it seems like everything in our genome, even the stuff we thought was useless, actually has a purpose. It's like a perfectly orchestrated symphony with every note, every instrument playing a vital role.
That's a beautiful analogy, and it's one that I think really captures the essence of what we're learning about junk DNA. But let's shift gears for a second. Let's move from the symphony to something a bit more dramatic, shall we? Because it turns out that junk DNA is also linked to disease.
Oh, wow. I hadn't even considered that. So how does this non-junk DNA play a role in disease?
Well, remember those regulatory elements we were talking about earlier, the ones that, control gene expression.
Yeah.
If those things get disrupted or altered somehow, it can really throw a wrench into the delicate machinery of the cell. And that can increase the risk of developing certain diseases.
So even though non-coding DNA isn't like directly making proteins, it can still have a huge impact on our health by influencing how our genes are expressed. Are there any specific examples of this connection between junk DNA and disease?
Oh, absolutely. Cancer is a prime example.
Right.
Researchers are finding that variations in non-coding DNA can actually influence a person's susceptibility to developing certain types of cancer. These variations can affect how genes that are involved in cell growth and division are regulated, potentially leading to, uncontrolled cell proliferation and tumor formation.
It's incredible to think that these stretches of DNA, the ones we once dismissed, could hold such important clues to understanding and maybe even treating diseases like cancer. It really highlights how important it is to look beyond that traditional gene-centric view of biology, you know what I mean?
Exactly. And the exciting thing is that we're really just scratching the surface here.
Mm-hmm.
As our knowledge of the non-coding genome expands, it's likely to revolutionize our approaches to diagnosing, treating, and even preventing diseases.
It's like we've been exploring this vast, uncharted territory, and we're finally starting to map out all its hidden treasures. Speaking of hidden treasures, I was really intrigued by the information you sent about pseudogenes. They sound almost like, like ghostly remnants of genes past.
They do, don't they? Pseudogenes are basically inactive copies of genes that have accumulated mutations over time, rendering them, well, incapable of producing proteins. It's like finding ancient ruins in your backyard, a glimpse into the evolutionary history of your genome.
So if they're inactive, why are they still hanging around in our DNA? Are they just taking up space?
That's a great question, and it's one that scientists are still trying to figure out. While pseudogenes may not produce proteins, they might not be entirely silent. Recent research suggests that some pseudogenes can still be transcribed into RNA molecules.
Wait, really? So even though they're broken, they might still be doing something. What could these RNA molecules be doing?
Well, there's some evidence to suggest that some pseudogene-derived RNAs might actually play a role in regulating their, corresponding functional genes. It's like finding out that those ancient ruins are actually emitting some kind of strange energy signal that's influencing the surrounding environment.
So even in their broken state, pseudogenes could still be influencing our biology in subtle ways. It just goes to show that we should never underestimate the complexity of our genome. Everything seems interconnected.
Exactly. And speaking of interconnectedness, there's another fascinating group of molecules we should discuss. Long, non-coding RNAs, or LNCRNAs. We touched on them briefly before, but I think they deserve a closer look.
Yeah, let's delve into those. Remind me again what they do. We've covered so much ground already. It's a lot to take in.
No worries. Think of LNCRNAs as like the master regulators of the cellular world. They're much larger than those tiny microRNAs we talked about earlier, and because of their size, they can interact with a wider range of molecules and have even more complex functions.
So if microRNAs are like post-it notes attaching to messenger RNA, would LNCRNAs be more like full-fledged instruction manuals?
That's a great analogy. LNCRNAs can act as scaffolds, bringing together different proteins that are involved in a particular process, or they can act as decoys, binding to molecules that would otherwise be interfering with cellular operations. They can even directly interact with DNA, influencing gene expression at like a fundamental level.
It's amazing to think that these molecules, which we once dismissed as part of the junk, are actually orchestrating such intricate processes within our cells.
It really highlights how much we still have to learn about the non-coding genome, and the potential implications are vast, particularly when it comes to understanding and treating diseases. LNCRNAs are actually being investigated as potential targets for therapies, aiming to manipulate their activity to combat everything from cancer to neurodegenerative disorders.
It's incredible to think that we might be able to harness the power of these non-coding molecules for medical purposes. It makes you wonder what other secrets are hidden within this vast, unexplored territory of our DNA. But before we move on, I want to circle back to something you mentioned earlier, epigenetics. I've heard the term, but I'm not entirely sure what it means. How does it relate to non-coding DNA?
Epigenetics is a fascinating field that explores how changes in gene expression can occur without alterations to the underlying DNA sequence itself.
So it's not about changing the letters of the genetic code, but rather about adding like annotations or instructions that influence how those letters are read and interpreted.
Exactly. Think of it like adding punctuation marks or changing the font style in a sentence. The words stay the same, but the meaning and impact can be completely altered.
Okay, that makes sense. So how does junk DNA play a role in this epigenetic regulation?
Well, some non-coding regions can be modified through epigenetic mechanisms, such as the addition or removal of chemical tags like methyl groups. These modifications can then influence the accessibility of DNA to the machinery that reads and transcribes genes, effectively acting like switches that can turn gene activity up or down.
So these non-coding regions are like the control knobs on a soundboard, fine-tuning the volume of different genes.
That's a great way to put it. And what's really intriguing is that these epigenetic changes can actually be influenced by environmental factors such as diet, stress, or exposure to toxins.
Wait, so our experiences can actually change how our genes are expressed without altering the DNA itself? That's mind-blowing. Does that mean our lifestyle choices could be influencing our health at a deeper level than we previously thought?
It's certainly a possibility, and it has profound implications for our understanding of how the interplay between our genes and our environment shapes our health and well-being.
Yeah.
It's like realizing that the story of our DNA is not set in stone, but rather a dynamic narrative that can be influenced by the world around us.
It's amazing how junk DNA has gone from being dismissed as useless to being recognized as a key player in this intricate dance between our genes and our environment. It seems like every discovery adds another layer of complexity to our understanding of the genome.
Absolutely. And it's a reminder that we should always be questioning our assumptions and be open to new possibilities. What we once labeled as junk might actually turn out to hold the key to unlocking some of the greatest mysteries of life.
Speaking of uncovering mysteries, I'm curious about introns. They're the non-coding segments within genes that get spliced out before the final protein coding message is produced, right?
That's right. They're often referred to as the bloopers of the genetic code, the bits that get edited out before the final cut.
But I'm sensing a theme here. If it's part of our DNA, it probably has a purpose, even if we don't immediately recognize it. So what are these bloopers actually doing?
Well, it seems that some introns might not be so useless after all. They can actually contain regulatory elements that influence gene expression. And in some cases, they can even be transcribed into functional RNA molecules that play roles in various cellular processes.
So they're not just passive passengers, they're actively involved in shaping the final product. That's amazing. It's like finding out that those deleted scenes from your favorite movie actually contained crucial plot points.
Exactly. And what's even more intriguing is that the presence or absence of certain introns can actually influence how a gene is spliced, leading to the production of different versions of the protein. This process, known as alternative splicing, adds yet another layer of complexity to gene expression, allowing our cells to produce a greater diversity of proteins from a limited number of genes.
It's like having a single recipe that can be modified to create a whole menu of different dishes. And all thanks to these bloopers that we thought were just useless filler, it really makes you appreciate the elegance and efficiency of our genome.
It certainly does. And as we learn more about introns and their functions, it's likely that they'll reveal even more surprises and challenge our understanding of how genes are regulated and expressed.
So we've gone from regulatory elements and non-coding RNAs
Yeah.
to pseudogenes, epigenetics, and even those intriguing introns. It's like we've peeled back layer after layer of complexity, revealing this hidden world within our DNA.
And we've only just scratched the surface. But before we delve deeper, let's pause here for a moment. When we return, we'll explore some of the bigger questions surrounding non-coding DNA, including its potential role in evolution and the ongoing debate about its ultimate purpose.
Welcome back to our exploration of the non-coding genome. It's incredible to think about how much we've already uncovered, from
Yeah.
those tiny microRNAs to those massive striptas of DNA that help shape the very structure of our chromosomes. But as you mentioned before the break, there's still some big questions surrounding junk DNA, right? Like its role in evolution and its ultimate purpose. So I'm eager to kind of dive into those a bit more.
Well, those are the questions that keep geneticists up at night,
Yeah.
And to be honest, there aren't any easy answers. But that's part of what makes this field so fascinating. We're constantly pushing the boundaries of our knowledge, challenging our assumptions, and trying to piece together a more complete picture of how our genome really works.
One of the things that really struck me in the research you sent was this idea that non-coding DNA might actually serve as a reservoir of genetic diversity, almost like a wellspring of evolutionary potential.
Yeah, that's one of the prevailing hypotheses right now, and it's a compelling one. Think of it this way. Imagine a sculptor with a block of clay. The genes are like the sculptor's tools, shaping and refining the clay. But the non-coding DNA is the vast, unformed mass of clay itself, providing the raw material for the sculptor to work with.
So it's not just about the genes themselves, but also about the potential they hold, the possibilities that are encoded within those non-coding regions. It's almost like having a genetic toolbox filled with all sorts of parts and pieces that evolution can then tinker with, combine, and even repurpose.
And that tinkering can lead to some incredible diversity and innovation. Over vast stretches of time, mutations and rearrangements within those non-coding regions can create new regulatory elements, alter gene expression patterns, like ultimately give rise to new traits and adaptations. It's mind-boggling to think about how those seemingly random changes at the DNA level can actually translate into the incredible diversity of life we see all around us. But it also raises another question. If these non-coding regions are so important for evolution, why don't we see them consistently conserved across species? Shouldn't those essential elements be preserved over time?
That's a great point. And it gets to the heart of the ongoing debate about the function and significance of non-coding DNA. While some non-coding regions are highly conserved, as we discussed earlier, others actually exhibit remarkable diversity across different species.
So what's going on there? Is that diversity simply a sign that those regions are truly junk, free to accumulate changes without any real consequences?
Not necessarily. Remember, evolution it's a balancing act. While some non-coding sequences might be essential for those core biological processes that are shared by many organisms, others might play more specialized roles. The adaptation of particular species to their specific environments.
So those non-conserved regions might actually be hotspots for evolutionary innovation, the places where new traits and adaptations are forged.
That's precisely. And that diversity might be what allows different species to diverge and thrive in unique ecological niches. Like each species has its own customized version of that genetic toolbox tailored to its own particular needs and challenges.
It's a beautiful example of how evolution can work on multiple levels, right? From fine-tuning those essential processes to driving the emergence of new forms and functions. But even with all this talk about evolution and diversity, there's still that nagging question of purpose. Is there an overarching reason why we have so much non-coding DNA? Or is it, as some have suggested, simply a byproduct of the evolutionary process, like a genetic echo of our past?
That's the million-dollar question, isn't it?
Yeah.
And as I said before, we don't have a definitive answer, but the more we learn about the non-coding genome, the more we realize that it's far too complex and multifaceted to be dismissed as mere junk.
You mentioned the selfish DNA hypothesis earlier, the idea that some non-coding DNA might just exist to replicate itself without any real benefit to the organism. Are there any other perspectives on this question of purpose?
There are. Some scientists propose that non-coding DNA might actually act as a buffer against harmful mutations.
Yeah.
It's like imagine having a protective layer of padding around your vital organs. Mutations are inevitable. But having a lot of non-coding DNA might help to absorb some of those random hits, preventing them from disrupting those essential genes.
So instead of being useless filler, non-coding DNA might actually be safeguarding our genome. That's a really intriguing idea. It kind of shifts the perspective from seeing it as selfish to seeing it as potentially protective. Are there any other theories out there?
Another idea is that non-coding DNA might actually contribute to the overall robustness and stability of the genome. It could be involved in maintaining the proper 3D structure of chromosomes, making sure that genes are properly organized and regulated. And some scientists even speculate that it might play a role in facilitating interactions between different parts of the genome, creating a complex network of communication and control.
It's like we're moving from a linear view of DNA, where genes are the sole actors, to more holistic understanding of the genome as this dynamic and interconnected system, with non-coding DNA playing a crucial role in orchestrating the symphony of life. It makes you wonder what future generations will discover about this hidden world within ourselves.
I have no doubt that future discoveries will continue to challenge our assumptions and reshape our understanding of genetics. And who knows, maybe someday we'll finally crack the code and unravel the ultimate purpose of non-coding DNA.
Until then, it's a journey of exploration and discovery, right? And I want to thank you for guiding us through this fascinating landscape. It's amazing to think that what was once dismissed as junk is now recognized as this vital part of our genetic heritage, influencing everything from our evolution to our health. I hope our listener will walk away from this deep dive with a renewed appreciation for the complexity and wonder of their own genome. Remember, there's no such thing as junk when it comes to our DNA. Every piece, every sequence, every molecule tells a story, and it's a story that's still being written.
What did you think of this week's episode? We'd love to hear your thoughts in the comments. Don't forget to join us next time as we dive even deeper. And as always, stay curious, because you never know what discoveries are just around the corner.